Okay, welcome everybody to the Plant Health Club. This is the second webinar we're holding. My name is Eric Bowe. I'm the Programme Secretary. I'm also one of the people contributing to this Living Science webinar. We've done these before at conferences, but now we have the opportunity to open it up to people anywhere and everywhere. I'm in London. Our speakers are in Shropshire and in Italy as well. So a varied menu ahead of us all. We're a small but vibrant and forward-looking society, as I hope you'll see after today. So these are career chats. This is less about the science and more about the pupil. The two are closely linked, and we'll be sharing our experiences, our path into science, and also our current job and some of the things that have affected the way that we've gone and influenced our career choices. So I'll briefly introduce everybody. Lucy Carson-Taylor is the head of plant health engagement at the Animal and Plant Health Agency, otherwise known as APHA. Lucy is based work-wise in York, I think I'm correct to say, and also works from home. Santa Olga Gacciola is an assistant professor in plant pathology at the University of Catania in Sicily. And I said, I'm Eric Bowe. You can find more about us on ResearchGate if you want to know more details. But let's come back to the meeting itself. We've all had different starting points and different ending points. We're going to be covering in terms of subject matter, plant health in general, what's often known as SBS, sanitary and phytosanitary regulations. That's the control and movement of plant material, plant pathogens and plant pests. I've worked a lot in tree diseases and plant pathology, and Olga has worked in Phytophthora, metagenomics and all that stuff that I struggle to understand. It is at the forefront of today's plant pathology. We will have 3 short interviews to begin the session, and that will introduce ourselves. Then we will be going into 3 separate rooms. But I think that's all I want to say at the moment, and just to hand over to Charlotte prior to starting the interviews themselves. They'll last for half an hour, 10 minutes each. We've pre-recorded those, so we keep to time. And then at approximately 20 to 2 UK time, we'll then go into the chat rooms. But I'll pass now on to Charlotte, who will explain how this is going to work in terms of the housekeeping and just general procedures. Charlotte, over to you.
Great, thanks, Eric. Good afternoon, everybody. So you will have noticed that you're all on mute at the moment. So just ensure that your microphones are on mute throughout the presentations, please. This introduction and the presentations are being recorded, but any discussions and questions after will not be. So please engage in the discussions without fear of it being recorded. There will yeah, as Eric mentioned, there's the pre-recorded presentations, and then we'll be split into breakout rooms. So Jenny is going to arrange this for us. So please, at this point, unmute yourselves and join in the discussions, or use the chat function. And there will be a facilitator in each discussion room who will make themselves known to the participants. So without any further ado, Jenny, would you like to start the presentations?
Good afternoon, Eric. How are you today?
Very well, thank you. All the better for seeing you.
Nice to see you too. So I just have a few questions to ask you today about your career. So how did you get into science?
Well, I had a decision made for me when I was at secondary school. I had a choice to go down an arts route or science route, which is, you know, I think it being very strange, but that was the way it was, and I couldn't make my mind up. So it was sort of semi-made up for me. Started into science, ended up doing physics, biology and chemistry. I dropped to physics and chemistry because I didn't like them, and went to university originally to do zoology, but then discovered I didn't like zoology very much because they were all interested in birds, and ended up in botany. And then just to finish this off, I discovered that I sort of liked plants, but I also liked microbes. So I put the two together and I got into plant pathology as my PhD topic.
Excellent. So what exactly was your PhD on?
I studied ash trees, which my mother thought was an ashtray, and she was quite willing to believe you could do a PhD in ashtrays. But I looked at a tree I couldn't identify before I started my PhD. I was slightly strange in that I had got my degree results first before I went round looking for PhDs. So I wasn't caught in that business of, will I get a good enough degree? So I went to several people, and I was rather taken by the person in Leeds. So it was purely a personal thing, a guy called Tom Priest, who's a really significant plant pathologist in my mind. He started physiological plant pathology, for example, which is now MPP. And I ended up in the agricultural building in Leeds looking at a bacterial disease of ash trees.
And so that PhD didn't put you off. So what did you do after your PhD?
Well, the thing that frustrated me like nothing was that it took so long to do artificial inoculation experiments, and that was an important part of my PhD. Particularly when you choose to inoculate your trees at the wrong time of year, which is what I did first of all. I couldn't even find ash trees to inoculate. I had to go to Askham Bryan College near York, and a very kind man there gave me his ash seedlings. So I did all that, and that to me sort of said, oh, you're now going to be a forest pathologist. So I looked for a forest pathology job, and there weren't very many around.
So what did you do?
I got to the fourth year of my PhD. Those are the days when you were only funded for 3 years, and decided that I needed to fall back on another qualification that I'd acquired, which was as a science teacher. So I went to Corpus Christi Roman Catholic Secondary School in Leeds and taught for 10 months whilst I waited for the government of Bangladesh to accept me as the world expert on bamboo diseases. This was a project, I should say, that was funded by the British government, part of the aid program. It's back in the 1980s now. So then what was then ODA, DFID, and has now changed again, selected me, but it took a little while to convince the Bangladeshis that a fresh person fresh out of the PhD was suitable. So I did the teaching at the end of my PhD, trying to combine that with teaching, which is the hardest job I've ever done. And then eventually, the end of 1981, I went out to Bangladesh to look at bamboo, disease of bamboo.
How long did you stay over there?
6 years, which all my friends thought was a bit mad. It was a lousy place to advance a scientific career in research, but a great place to learn about development, which is what I've subsequently been involved with. So there until 1988, including the arrival of 2 children. So went with none, came back with 2, and then started to look for another job. So that was bamboo blight for 6 years. And then I waited for the next disease to come along.
And what was the next disease?
Oh, I go from one not very well-known plant to an even more obscure plant, which is clove trees. When I say obscure, these are incredibly important. 600,000 hectares, I seem to remember, in Indonesia. And now another bacterial disease, so I sort of that in my armory of experiences to work on. And I was there for 2 years on a project been going for a long time. A lot of excellent scientific work had been done on that by people like Chris Lomer, Simon Eden Green, and many Indonesian counterparts, Supriyadi, Rodia, and many others. So that lasted for another 2.5 years. So 1991, I'm a sort of forest pathologist, but know nothing about trees growing in Great Britain. So come back to the UK, and then what? Not quite sure what next.
What did you do next?
I then got phoned up by Mike Jager at the then newly formed Natural Resources Institute, and they were looking to fill up posts in pathology. I then became the tree crops specialist, and you can see the tree theme keeping on going, and stayed there to 95, by which time it was slightly falling apart because an institute that originally had 400 people went down to, I think, about 80, and then I joined CABI. So that's the Centre for Agriculture and Bioscience International. I joined them with the help of Jim Waller in 1995 and stayed there until 2012.
So you must have really liked it there.
Well, I did. I mean, I think my problem has been I was desperately looking for a home to work in, and that's what CABI gave to me. So they gave me a lot of freedom. So I then didn't quite cast aside the tree diseases, but moved much more into plant clinics and into extension, and got back to teaching, which is what I always liked doing, but I just didn't want to have 32 reluctant teenagers in front of me having to study for their GCSE in biology. So the CABI gave me that huge freedom, but also having to find money to do work as well. And that I realized from early on I had to work hard at that if I wanted to do things that really engaged me, like the plant clinics. So that really took up a lot of my time. But I also did other things like looking at wild mushrooms, which is might sound a bit strange, but it has a development theme. It's about people, you know, and there's even included cucumber viruses as well, I believe.
Oh, yes, that sounds familiar. What would you say is the highlight of your career?
I think the highlight career is still enjoying myself at the age of, you know, from 50 onwards, when I really knew what I wanted to do, which was a mixture of natural and social sciences based on plant health. So it's not a single, but I can pick out certain points, but it was just being able to do things that allowed me to be curious, to work with people, to set challenges, and to come up with achievements, be it publishing a paper or just sitting in the back of a truck high up in Bolivia, watching somebody do an impromptu explanation of nematodes and potatoes. I mean, just absolutely a thrilling event. So I got a series of these along the way, but it was the flexibility and still working at a late stage in my career and setting new challenges.
That sounds wonderful. What advice would you give to the younger generation based on your experience?
What advice? I suppose one of the things that's mattered most to me is having good colleagues and collaborators. And when you find them, do everything you can to keep on working with them. I've worked with many different people, but there are several with whom I have a special relationship. One is an anthropologist. His name is Jeff Bentley. We've written lots of things together. And then Jeff and I worked with a lady called Solveig Danielson in Nicaragua, and before that and after that. And these are people you can utterly rely on. These are people who challenge you as well, by the way. It's not meant to be a jolly when I say colleagues, but you know, when they become close friends and when they become good colleagues, that's what makes your life worthwhile as a scientist, because you cannot do this by yourself.
That's very good advice. Do you have any choices that you regret?
Well, I suppose I could regret going into tree diseases because I thought it was a dead end. You know, you're sitting there, and this is not meant to sound flippant or facetious, but you're waiting for the next big disease to come along. And trees just don't get diseases very often. It's not like working on rice and maize with our constant channels, or vegetables, or fruit trees. You know what's there. But I think it's just to keep on going, to write and write again, you know, and to get out there and mixed people. But so I'm drifting away, as I often do. But what I regret, I don't think I regret anything. In retrospect, I think at the time I felt there was bad decisions, like why did I go to Bangladesh for 6 years and sort of go into this scientific cul-de-sac? But in retrospect, there were many aspects of that which I'm really grateful for, like learning about the context and the countries and development, because you cannot do science overseas unless you are pretty resilient. So, plus I hated at times driving in horrible temperatures in a non-conditioned air-conditioned vehicle. In retrospect, I see that as being a good thing. So I don't think I've made bad choices because I'm still enjoying doing what I'm doing now. So it's all worked out eventually in the end.
That's brilliant.
So welcome, Santa Olga Cacciola, and to this Living Science. It's an opportunity for us to learn about each other as scientists and our careers. So we've known each other for some time now, and I thought it would be interesting for people who are on this webinar to learn more about people's path into science and some of the events along the way. So my first question is, how did you get into science? What was the starting point for all this?
Okay, it was long ago, I thought that I could tell a story about that because I think it was important for me. When you asked me this question, when I read the question, I immediately something got back in my memory. Okay, I think 3 important events were crucial for my decision to go into science. 2 of them took place in my high school, and the third one at university when I was at the Master of Science.
Right.
The first one was a bad experience, really a bad experience, because my professor assigned as homework the description of the structure and function of a cell, of an eukaryotic cell.
Mm-hmm.
And I was in my first year and I really didn't know. I never heard about cells. So I said, what to do? I just learned by heart and I was cut. They cut because she asked me to talk about cells. So I just learned by heart. And I got my first really bad score in my life. So I was really upset, but for me, it was really a lesson. Okay, it was my first lesson, but because I was the first time, I just went into a cell. I just bumped in something like cell, and then okay, this was done. The second experience was always in high school, but the third year, so I knew a little bit more. And I got a professor, I still remember her name, and the name was Professor Reina. She was really a very bizarre person. There were stories, going around about her private life. She had this strange way of talking, but anyway. Above all, she was very grouchy and inflexible, and she was teaching biochemistry and plant biology.
Mm-hmm.
In plant biology, including all the metabolic pathways that are very difficult to learn, to understand. But I was so fascinated by this hidden and perfect cellular world where all the metabolic reactions are performed and allowing all the living organisms to be alive. So the research about unraveling the molecular mechanism of living organisms started to interest me and stimulate my imagination because I started to see all the tubes and how you can see the inside there are cells. But the third most fascinating experience was at university. I was at my, I think, second or third year, I don't remember. And I fell in love with plant biochemistry and the study of fungal diseases. Because I don't know, for some reason they captured my fantasy. The idea of working with in a laboratory with steels and test tubes, and to understand the invisible mechanism undertaken inside every living organism really thrilled me. And then I decided this would be my life. And at that point, I took my decision. So what I did after that, well, something happened.
So when you finished your master's, did you know then you wanted to do a PhD? You found
No, okay, no. My story is very complicated. I'm making it very simple, but I had a complicated story in this sense, because soon after my master's degree, I went abroad. Okay, at that time was very difficult that Italian girl, Sicilian girl could, even think to go abroad. But for me was I was, and not only my father. I remember, we're just a parenthesis. My father wanted me to become a gym teacher of, how do you say, gym teacher, gymnastics teacher. Yeah,
gymnastics teacher. Okay, right.
Yes, because he said he was a head of a high school, and he said this is the most simple job. You don't have to leave to work a lot. It's not heavy. It's simple. It's nice. But
Right.
of course, I said no.
So
Anyway,
it's curious because many parents have high aspirations for their children, but your father had relatively low aspirations.
Because he thought that he was very he had a very hard life, and he became head of the school after doing the war. I mean, really hard life. And then he became almost a university professor, but then the story is different. And then at the end, he became a professor at a school, and then a head of the school. So I think he thought that it was going to be very difficult, path and all the problems. And this was his advice. But anyway, I didn't, of course, listen.
So where did you do your PhD?
Okay, what happened? I mean, after my master's degree, I went to work abroad at the University of Bern in Switzerland. And there I studied transport mechanism in red cells. Then I went to the States at Johns Hopkins, and there I studied adhesion and communication cell mechanisms with a very brilliant scientist, really brilliant scientist. When I came back to Italy, because I had to come back to Italy after a long stage in the States, I was very happy to catch the opportunity to continue to do research in molecular biology applied to plant pathology. And there the story started, because I started the Faculty of Agriculture at the University of Catania, and I started my research in a field that was quite new for me, which is the possibility to apply biochemistry, my biochemistry skills, to the field of plant pathology. Okay, then I started my PhD. So I did my PhD in plant pathology because I decided to work in this field. Okay, and to do my PhD, I didn't do it not only in Catania, but most of the time I was in Bari. And as maybe you know, that University of Bari is one of the best in this field, in the plant pathology field. And there professors such as Ciccarone, and I don't know if you remember, if you know him, Antonio Ciccarone, and his pupil Antonio Graniti were working. Antonio Ciccarone was already dead, but Professor Graniti was there, and I had the opportunity to work with him for a short time. It was a really witty and such a strong intellect and a deep knowledge of and visionary view of the discipline. And so I was really fascinated again and more and more. And I think that this period was crucial for my education, but also for my career as a young researcher, and I gained the confidence to continue my career in this field. So this was my story with plant pathology.
So you've moved from Catania, which some people won't know is in Sicily, it's across the Straits of Messina, up into Apulia. What about
Apulia.
cultural differences? Did you just go to Apulia and it was like being at home, or was it quite different?
Oh well, Apulia is a beautiful region. I was in Bari, so I was at the capital. And Bari is nice because as these differences between, you know, the area like a big town, but then there are part of the town which are forbidden for most of the people because they are criminals, no?
Okay.
But I remember that I was used to go with other friends at night, working there. And I never felt, you know, I never felt really, come si dice?
In danger.
Right, danger. I felt always like a nice town, wonderful food. They have wonderful food, but also wonderful vegetables.
Right.
I was living in a street just above the open market. That was almost every day because I didn't have much money, you know. So I had to run a place very economic, but I was very near the university, so it was very comfortable. And I remember all these smells, good smell, but actually I didn't cook a lot, so I didn't take profit of this market.
Time-consuming thing doing a PhD. So tell me just briefly then where you are at the moment and what your current jobs are. What are you studying? Because you've come to plant pathology through biochemistry, through physiology, and now you're in plant pathology. So
yeah,
a little
right.
bit about that.
So I thinking about this answer, I think that I face different challenges in plant pathology. I started as a researcher and I applied for the first time, thanks to my experience, gel electrophoresis of proteins and isozymes for the diagnosis of Phytophthora species of agricultural interest, of course. And for that time, it was an innovation, at least in agriculture. And because the DNA era was approaching, but was not still there. So I would say that this innovative and pioneering approach often made the opportunity to become and then be recognized as internationally as an expert in Phytophthora species and diagnosis of plant diseases. So I think this was something for me. And the other thing I feel to say is that I've encountered many difficulties. I was staying in Palermo for 10 years with my little babies, but my enthusiasm for the scientific world has really never faded. This is, I think, what is the clue of my.
Yes, I mean, you have to be determined to succeed, because I know that in some countries in Europe, trying to get to the top, you have to struggle, particularly, I have to say, as a woman.
Right.
But you have got there, and one thing we will talk about later on during the breakout sessions is something about that all. So that's a great introduction towards all this. Olga, thank you very much indeed, and we look forward to continuing these discussions in the rooms with the participants. Thank you again.
You're welcome.